Thank you, Dr. Delmonico. It's a great honor to be here with everyone today. As Frank mentioned, I was asked to talk about the current state of organ transplantation, but will, I think, focus more on what's coming in the future. The last 20 years or so of organ transplantation has been relatively stagnant without any major breakthroughs, but there are many new technologies coming online that have the potential to really change what we do. So I've listed 3 of them here that I'd like to mention each briefly. The first is mechanical preservation and assessment of deceased donor organs. The next is stem cell-derived transplants for diabetes, following on Dr. Blau's comments. And the final is xenotransplantation. So one of the main limitations we have in transplantation is a period of ischemia that all the organs suffer. So this is from the time the organ is removed from the donor and transplanted in the recipient. And the standard is called cold storage. The organ is placed in a box on ice to slow the metabolism. But we know that being without oxygen and nutrients and blood, the organ suffers during this period. And this can result in poor function in the recipient that sometimes can be life-threatening. Having the organ in the box also doesn't permit any optimization of the organ, and it doesn't really permit assessment of function or viability. This can lead to poor results and poor utilization of organs. This, we think, is the solution. These are the 4 main organs that we transplant, each being preserved ex vivo with a mechanical device. Each is being preserved physiologically at body temperature. The lungs are functioning to oxygenate blood in the circuit. The heart, you see, is vigorously pumping and gives some sense of how well it'll function when transplanted. Liver making bile, detoxifying the fluid in the circuit, and the kidney making urine. So this takes care of most of the issues that I just mentioned that we face with preservation. This shows that a little more clearly. So some organs are damaged in that they have a period without blood or oxygen in the donor. This leads to consumption of all their energy stores, shown here as ATP levels. Once the organ's put on the pump, within a couple of hours, 2 to 3 hours, the ATP levels are back to normal. So the pump can actually make damaged organs better. We also can use it to assess organs. When we recover a liver, we judge it often based on its appearance and some blood tests prior to recovery. But we really don't know whether it's a good organ until we transplant it. The pump helps us look at that and the physiology of the organ, shown in the kind of bottom right box. This measures lactate clearance by a liver on the pump. A good, well-functioning organ rapidly clears lactate. The red lines are a composite of about 100 livers that we put on the pump. The 2 gray lines up top are 2 organs that didn't clear lactate and would have functioned very poorly and could have put the recipient at risk. So the pump may also weed out organs that could be damaged or dangerous. Probably the most important benefit of using the pump would be to assess some of the organs that are currently discarded. There's thousands of organs each year that are recovered and not used for various reasons, mainly because surgeons aren't certain whether the organ would be suitable for transplant, and if there's any doubt, they're apt to not use it. So the pump should make more organs available for transplant and save lives. There's also many new things that we can do with the pump that are just being explored now. You can imagine having a liver or a kidney on the pump for 24 hours. We may be able to induce genetic modifications in the organ that prevents rejection or inhibits inflammation. And there are many others that are currently being studied. So the pump is a powerful device that can be used for new advances that are just being explored now. So the second topic is the treatment of diabetes with stem cell-derived islets. For the last 20 years or more, we've transplanted islets from deceased donor organs. So recovery of the pancreas at the time of organ recovery. We take the pancreas to the laboratory and we digest it, isolate the islets, and then infuse the islets into a diabetic recipient, usually into the portal vein where they lodge in the liver and function normally. Unfortunately, the process is very inefficient. In general, the preparations we make only have about 30% islets. The rest is contaminating exocrine tissue. The organs that we get are of variable quality, and it may take up to 4 processings to get enough islets to reverse diabetes in a patient. So a very inefficient process. But the biggest issue is that the deceased donor organ supply is wholly inadequate to serve all the diabetics who could potentially benefit from beta cell replacement. So we think the solution may be in stem cell-derived islets. Many groups around the country have worked on differentiating stem cells into cells that produce insulin or that develop a full islet with multiple cell types. Some of the best work came from Dr. Melton at Harvard, who was able to fully differentiate stem cells into islet-like clusters and show that they functioned in mice and functioned in vitro to release insulin upon glucose stimulation. It's a complicated process, may take a month to do, and 25 different factors have to be added at precise time points to bring about this differentiation. Because of those facts, it had to be transferred from the laboratory to industry. And Vertex, a pharmaceutical company that you all know, took this over and developed this into a clinical product. The product is called VX-880. You see on the left, those are a beautifully pure, perfect preparation of islets. The islets, these cells are also delivered into the liver where they lodge and function. Because they're foreign, the recipients have to take immunosuppression. And I want to tell you about the first patient that received this therapy about a year ago. So this was a 64-year-old male who had type 1 diabetes for 42 years. And with time, his disease got more and more difficult to manage. He had poor control, as seen by his A1C, and no detectable islet function at all based on his C-peptide. The biggest problem was that he lost the ability to sense when his sugars went low. And this caused him to become confused and sometimes go into a diabetic coma. And that happened about 5 times in the prior year. Previously, he had a motorcycle accident when his sugar went low, and he worked as a mailman, and on his route, he would be found down on the ground, confused, not where he is when his sugars drop. So a very debilitating disease and really a great candidate for a new study like this. So we have lots of data looking at how he did post-transplant, but I think this slide here really encompasses the key features. So this is what's called continuous glucose monitoring. He wears a sensor on his arm that monitors his glucose every few minutes. And this is recorded over a 2 to 3 week interval prior to transplant on the left, and then at about 4 months and 8 months in the other 2 panels. The target glucose range in which all of us, or most of us, live here would be between 70 and 180. That's the gray shaded area. But you can see his glucoses had wide excursions from lows below 50 to highs above 400, and this was happening every day and was just uncontrollable. He was only in the target range about 40% of the time. By 4 months post-transplant, his insulin requirement had dropped by 90%, and he was in range now 80% of the time, so markedly improved. And when you see at 8 months post-transplant, he's now completely off insulin, almost 100% within the normal range, and with an A1C of 5.2, really quite a profound result. And even though this is only the first of these that have been reported, it really shows the potential of stem cell-derived product in this area of diabetes. I thought it would be worth hearing from the patient in a little news clip that was recorded a few months back to see really how he suffered with this disease.
64-year-old Brian Shelton has lived with type 1 diabetes for more than 40 years. 15 years with dangerous episodes of hypoglycemia.
You spend your entire life, every minute of the day, trying to keep track of where your numbers are at. Did I eat? Did I not eat?
Sometimes when his blood sugar dropped, he says he'd lose consciousness without warning.
I shoveled down the snow, and when I got inside the garage, my number crashed, and I went face first into the concrete.
But now Brian's life has changed. after becoming the first participant in a groundbreaking new clinical trial.
How do you feel right now?
Right now, great. Just fantastic. I feel like I got released from handcuffs. I'm not sitting here like, where's my orange juice? Where's my pills? Where's my you know, it's like, I don't need them.
Brian says he can now go days without taking insulin and no longer fears those sudden, dangerous crashes.
Now I could do yard work without having to worry about my numbers going too high or too low. All my numbers are right in that line. And just seeing that, you know, makes me feel great. I feel like I'm getting another chance at life.
Hopefully there'll be many others like Brian that come along, but I think it really shows what the potential is here. Now, these patients do have to take immunosuppression, so there's a balance, and it's only appropriate for those with very labile, severe disease like Brian has. But there's much work going on in the community to try to genetically modify the cells so immunosuppression is no longer required. And I think that if that does succeed, then they could be used in essentially any diabetic. So I'm going to switch gears to the final topic, and that's xenotransplantation. So this idea has been around for a long time, using animal organs since we don't have enough human organs. In the 90s, there was a really concerted effort. Industry was working hard to bring animal organs forward. But the modifications were difficult to make. The field was moving slowly. It was costly. But what really shut down the field was fear of transmission of a virus from pigs to humans. And that one specific virus was called a porcine endogenous retrovirus. And this really shut down the field at that time. This led to essentially every meeting that one would go to about xenotransplantation would use this quote, that xenotransplantation is the future of transplantation and always will be. And that skepticism now is gone. And it's gone because of the new technology that allows a more easy editing of pig donors to induce modifications that will make the organs more suitable for transplant. And of course, that's a development of CRISPR-Cas9, for which Dr. Charpentier and Doudna received the Nobel Prize a couple of years ago. So this was taken on by a couple of researchers at Harvard, George Church and Luhan Yang, who started a company to make pigs more human through genetic modification. And the first they took on was this endogenous retrovirus. And using CRISPR, they knocked out all 62 copies of this endogenous retrovirus in the pig genome in one step. And prior to that, the most number of modifications done at one time was only 2 or 3. So this showed the real power of CRISPR compared to the existing technology and opened the door for more modifications in pigs. But also now we have pigs that we believe are safer, even though no one really knew what the risk of the PERVs was. So there's other impediments to using animal organs. We all have antibodies against antigens that pigs express. These have now been knocked out on the pig to get over that barrier. And there's other physiologic barriers. I won't go into much detail, but complement and coagulation and immune inflammation are kind of disrupted when pig and human pathways work together. So human genes are being inserted to address these pathways to get over that barrier. And you can see on the right, the team at MGH has been working with eGenesis, headed by Dr. Kawai, and over time, the results have become better and better at getting pig kidneys to survive in non-human primates as a test for humans. This shows where we are overall. There are kidneys now that have survived more than 3.5 years in monkeys, pig kidneys. Hearts that have survived for 3 months if they're put in a life-sustaining position. And the experiments were terminated at 9 months per protocol. They would have lasted much longer. The 2 problem organs that remain are lung, only lasting to 31 days, and liver to 28 days. Unfortunately, liver is the area in which I work, and it's been a very difficult model, but I think we are making slow progress. And it's possible that liver may still have a clinical purpose, perhaps combining with one of these perfusion pumps to work as an ex vivo support in patients who are dying of fulminant liver failure. We think the liver could be used sort of as a dialysis unit, but for liver failure, and help these patients. The other major advances or developments of late in xenotransplantation was transplanting pig kidneys to deceased humans. So these are patients or individuals who expired, were legally brain dead, but weren't able to donate their organs, but the family willed the body to use for scientific research and specifically this trial. And the team at NYU has now done 3 patients, 3 decedents who received a pig kidney transplant that was monitored for about 3 days to see how it would function. And they functioned very well. This was reported in New England Journal back in May. Kidneys function well. Histologically, there was almost no evidence of rejection or a problem, and made the point that this can potentially be applied in humans. And then finally, that I'm sure you all heard about, was the first pig human heart transplant. This was a 10 gene edited pig that survived for 3 months in a desperate patient who didn't have any other options. So tremendous things happening in xenotransplantation. And certainly I think xenotransplant has arrived, and within the next 2 years, we'll see concerted trials, probably for kidney and maybe heart. Thank you.
So questions from our academicians or participants this morning, please. Yes, Professor Chen.
Thank you very much for your talk. I would like to ask you a question is, what are you going to do to expand this kind of innovation to other institutions, or how to benefit the more patients?
That of course, is the key question. And I think that each of the technologies may be a little different. The pumps are very expensive and very manpower dependent. But I think the other technologies of islet transplantation from stem cells should be more available and probably cheaper in the long run than what we do right now, recovering 4 organs and processing that each time costs $100,000 or something. So I think the stem cell technology should derive a cheaper product that is more widely available. And I think probably the same is true for xenotransplantation. Right now, we go to tremendous efforts to go recover organs. 3 different teams may fly, 5 hours away to recover a single organ. If we had pigs that were donating organs that could be shipped to those areas in need, it would, I think, be much more economical and much more feasible.
Professor Blau and for Professor Markmann, let's think on this question together, perhaps. Jim, your patient required immunosuppression, so that the islets were derived, they're not autologous. But what is the opportunity to take a stem cell from the person that is in need ultimately of the revision, the transplant, the improvement, and have those stem cells in the midst of the transcription factors come along to have a sufficient quantity that can impact their health without the requirement of immunosuppression? So perhaps Jim could answer that, but then we'd like to hear from you as well, Professor Markmann, your answer or thoughts on that.
Yeah, that's a great question, and certainly there's great interest in this. At least right now, I think the impediments are largely regulatory. So scientifically, there would be no problem or difficulties in doing that. But each individual's preparation of iPS-derived islets would have to go through a full FDA approval process, since they're all different, and that costs millions of dollars. So I think that'll eventually change with time, and they won't all be recognized as different products. But until then, I think the main hurdle is an economic hurdle. Maybe Dr. Blau has other thoughts.
Yes, that's the point I would make, that it's extremely costly to grow enough stem cells and to do it in personalized medicine, where you take a person's own cells, either the tissue-specific or the ones derived from pluripotent cells. It would be very expensive to grow up enough for each person. So finding ways to get around the immune response would be huge, because then you could have a supply that could serve many, many people.
To be in Madrid the other day and be involved with the European Commission, and the concern of the European Commission as it pertains to tissue transplantation, medical products of human origin, stem cells, and then the evolution of stem cells and the cost of their proliferation. As Professor Markmann mentioned, this has been taken on by industry. And the concern of the Commission is, and this goes to Professor Chen's issue of widespread distribution, industry now takes over that project because it's so expensive. But then industry is wanting a compensation. And the concern becomes, in widespread distribution, that the poor person that can be the source of these stem cells, that can be then subsequently proliferated, becomes quite a billion-dollar industry. So, Professor Blau, perhaps your comment about, in the regulation of this, that industry is going to take it on, the compensation that seems to be forthcoming, and whether or not that compensation will enable its widespread distribution and impact public health.
It is a dilemma, because in order to bring a drug to the clinic, you need industry. You need that toxicology, quality control, all the FDA requirements. Almost impossible to do that in a lab. We don't have the support in a university to do that sort of thing. We're not trained to do it ourselves. It requires a lot of expertise that pharma has. And so that's the dilemma. You need them to do that. Their motivation is, as you say, often mixed. It's for the benefit of people, but it's also compensation. And I think we need to work to make it more broadly available, and we need to have policies from the government and from the NIH in order to try and change that, to make these accessible, so that I guess the first test will be quite expensive, but then ultimately it could be made available to people without waiting for the patents to become superfluous, which is what happens now.
I'm curious to know what you think about these pumps. Is this just oxygenation, or is there a beneficial effect of recapitulation of mechanics per se, of the movement, of the contraction? I imagine, for instance, for heart, this must be very important, but in the future for lung, are very delicate, this may be key per se, irrespectively of oxygenation. What is your feeling?
So I think there are, I think it probably is both, that the oxygenation is clearly what has changed things. But we do know that when the organ is on the pump, the vasculature is often very constricted. And I think both the pulsatility and we sometimes administer vasoactive substances to vasodilate are both very important in getting the organ well perfused. So I think the pulsatile nature of the pump, I think, is key as well.
Next introduction, but as I do, President von Braun, this academy may be impacting at some point the regulatory aspects of what it is that the industry, the pharmaceutical industry, comes to bear upon its compensation, and yet the ability to have it widespread distribution and impact health. The academy hopefully can have some impact.
Well, let me echo that hope. We are committed to stay on the message, to see the opportunities of organ transplant activities and the dangers of organ trafficking as part of human trafficking. We are committed to that and look for opportunities of science to solve both.
Okay, let's move to the third speaker. The third speaker will be given by Dr. Stanley Brusner, who is the director of the Institute for Neurodegenerative Diseases and professor of neurology and biochemistry at the University of California, San Francisco. He's going to talk on the alpha-synuclein prion strands as the causes of dementia with Lewy bodies and multiple system atrophy. Professor Brusner, the floor is yours.
Thank you.